better or not?
Yes,
Much better.
that's good.
Okay, great. One more thing, can you see my cursor or not before I start? Is there a cursor moving around or? No?
Yes.
Oh, yes, there is. Great, thank you. Thank you for the kind introduction, Enrico. I'm very happy to be here and share the research that I and people in my lab have been doing. And I want to go right into it. Let's see. Why is it not switching? Oh my God. For some reason, okay. Yeah, now it's switching. Great. So I have a couple of things for you on the menu for you today. First, I'd like to introduce insect-specific viruses and mosquitoes.
Described.
These are viruses
Describe.
people don't generally study on, or at least it's not the first
Dingo.
viruses people think about when they think about viruses and
Doorframe.
mosquitoes. So I'd like to introduce them and talk about how we're doing
Dragon.
research looking at these insect-specific
Del
viruses.
Camera.
The second part of the talk, I want to
Durian.
give a brief overview of virus discovery I've been doing in the Netherlands in Culex
Eared.
mosquitoes, where I
Ear.
had the opportunity to look where people had not been
Echidna.
looking before. So we had no idea what we're going to find. So I'd like to spend
Echinoderms.
some time to talk about this.
Echinoderm.
However,
Egret.
the larger part of the talk will be about a global
Elk.
mosquito virome database that I've set up together with two of my PhD colleagues, where we really we made a database compiling all the information that people had found regarding insect-specific viruses and mosquitoes. And when compiling all this information, we wanted to see what we could now say that we know about this mosquito virome. First, I want to briefly introduce why we study mosquitoes in the first place. Here we're showing you three representative mosquito species from three different mosquito genera, which are all quite critical with regards to pathogen transmission. And this is really one of the main reasons researchers have been studying mosquitoes, is because of their ability to transmit both viruses, but also parasites like malaria to humans and other vertebrates. I also have a picture of mosquitoes in action in three different ways. For pathogen transmission, we're mainly talking about blood feeding. And this is also what people generally think about when it comes to mosquitoes. However, people tend to erroneously assume that this behavior, this blood feeding behavior, is mainly for nutritional benefits. Whereas actually the picture in the middle here, a mosquito on a flower is probably much more representative of a mosquito feeding. And that is because both female and male mosquitoes, they require nectar to survive. And it's actually only female mosquitoes that require blood meals to get protein that they need to develop their eggs for their offspring. And because of this nectar feeding, mosquitoes also play a role in pollinating plants, and they also play an important role in the ecosystem as food for various animals, such as birds, but also fish or reptiles, amphibians, etc., and other insects, actually. However, for some reason, what you might not know is that mosquitoes are also very photogenic, as indicated by this picture, which for some reason, some researchers might be good enough reason to want to study them. However, I'd like to talk to you about arbovirus transmission. Arbovirus is short for arthropod-borne virus, and these are viruses that not only have a mosquito host, which is an arthropod, but also have a vertebrate host, which in this case would be humans. And the main point I'd like to make here is that this is really a transmission cycle. And mosquitoes do not pick up a virus from the wild. They're not born with these viruses. They have to pick up an arbovirus from an already infected vertebrate that has these viruses inside their blood. And then subsequently, if a mosquito were to transmit this virus to a next host, it would have to take another blood meal from an uninfected host and transmit the virus in this way. And this transmission cycle looks approximately like this for viruses such as dengue virus, yellow fever virus, and Zika virus, which are all very well-known arbovirus in the world. However, this transmission cycle can look different for other arboviruses, such as, for example, West Nile virus, where the main host for transmission of these viruses is actually bird species. Where humans do get infected, they also can develop disease in a small percentage of cases, but the infection is usually not strong enough to be able to sustain a transmission cycle. In our lab, we are mainly focusing on the mosquito side of things, but basically focusing on this step. What is happening inside of the mosquito? So I want to zoom into this. Here you have a depiction of a mosquito taking an infectious blood meal from a vertebrate that has these viruses in viremic in their blood. So this virus is picked up, this arbovirus is picked up, it comes into the gut system and reaches the midgut, where it needs to infect cells lining the midgut and subsequently escape the midgut barrier. Having escaped the midgut barrier, it enters the hemosol, which is the blood-like fluid of the mosquito, from where it can disseminate into a whole range of other tissues, with the ultimate goal of having enough virus particles in the saliva of the mosquito, so that when a mosquito takes a next blood meal, and when it does, it will actually excrete a bit of saliva, there are enough virus particles there to infect this new host. The key point I want you to take away from this is that mosquitoes are therefore not a passive host. These viruses need to actively replicate and effectively replicate inside of this mosquito in order for them to be transmitted, which makes arboviruses really interesting because they have both an insect host and a vertebrate host where they need to actively infect. The second point is that any factors that influence how well an arbovirus is replicating inside of this mosquito would affect how well this arbovirus is being transmitted by the mosquito. And this is where we come to insect-specific viruses. As opposed to arboviruses, mosquitoes also carry a whole range of insect-specific viruses. And these insect-specific viruses, their transmission cycle is usually assumed to be quite different. There's no vertebrate host there. And although this is a simplification, it tends to be assumed that these viruses persist in mosquito populations through what is known as vertical transmission, where mosquitoes, either male or female mosquitoes, via infecting both sperm or eggs, infect new generation in this way. Because at this stage, the arbovirus is replicating inside of the mosquito, and there's also insect-specific viruses there, they can interfere with how well this arbovirus can replicate inside these mosquitoes. And there's some initial evidence to suggest that some of these viruses can indeed affect arbovirus replication. The mechanisms for this are not quite clear. It could be that the presence of both an insect-specific virus and an arbovirus in the same cell could affect arbovirus replication through competition for the same host resources, or perhaps insect-specific viruses prime the immune system in such a way that arboviruses would not replicate as effectively. So really this effect of insect-specific viruses on arbovirus replication is one of the main reasons that people have been studying insect-specific viruses. One of the final point I want to make is that when I'm talking about the virome of mosquitoes, subsequently in this presentation, I specifically refer to this whole range of insect-specific viruses and purposely exclude the presence of arboviruses, which could definitely be considered part of the virome, but this is how I'm going to use this term. there are several different strategies that researchers have been using to characterize the mosquito virome. And I think you're all quite familiar with them. So the main method, at least recently, is using either RNA-seq or DNA-seq. And the advantage of this technique is that you can pick up all the RNA inside a mosquito sample, and therefore you have a very unbiased method to look for viruses. The downside of this method is that you are able to pick up all these virus sequences that you may not be so interested in. So apart from insect-specific viruses and arboviruses, you can also pick up virus sequences that are infecting pathogens of the mosquito, symbionts of the mosquito, viruses that were present in blood or nectar of a recently fed mosquito, or virus sequences derived from EVEs. And EVEs is short for endogenous viral elements, and some specific mosquito species, the genome seems to be ridden with these endogenous viral elements, which are integrations of viral parts inside the DNA, which can be transcribed and picked up by RNA-seq as well. So although you can pick up all these sequences, it becomes quite difficult to distinguish the ISVs from all of these other sequences. Another method that people have been using to find insect-specific viruses is using PCR screens, either using family-specific virus primers or specifically looking for a certain virus. And of course, the advantage here is that you can test a lot of different samples very easily and very cheaply. The downside, of course, is that you can only look for the viruses that you are specifically interested in. One of the ways in which you can better prove that you have found a virus that is really an insect-specific virus really replicating inside of a mosquito is by using culture approaches. So what you can do is you can homogenize mosquitoes and put that on an insect cell line, such as this, and allow any virus to grow out inside the cell line. And if you have a virus growing out inside a mosquito cell line, this supports that it's likely to be that these viruses are also actively replicating inside of this mosquito. The downside, however, is that you're selecting in many different ways. You're selecting for viruses that replicate inside your specific cell line that you choose. There might be competition if you have multiple viruses trying to grow out. Some might grow out better than others. But also, usually we're looking for visual signs in these cells that there's a virus replication happening. And some viruses might not give these visual signs and will therefore be missed. Finally, I want to talk a little bit about small RNA-seq, which is basically like RNA-seq, but then specifically sequencing the 18 to 30 nucleotide range of RNA. And this has some advantages. This allows you to solve some of the issues that full RNA-seq might give, but it does require a little bit more background. One of the cornerstones of the immune system, specifically the antiviral immune system of mosquitoes, is RNA interference, also short for RNAi. And the idea of this defense mechanism is that when a virus is actively replicating inside a mosquito cell, it will create double-stranded RNA as a replication intermediate. This double-stranded RNA will be detected by dicer2, which will cleave it into small fragments called small interfering RNAs, which are specifically 21 nucleotides long. These small interfering RNAs can then be loaded into 802, which, due to the sequence similarity, can find and subsequently cleave viral RNA. This defense mechanism seems to be very broadly effective, and therefore small RNA-seq works on detecting these 21 nucleotide sequences and finding actively replicating viruses through this method. So one of the important publications showing the benefit of small RNA-seq for this approach is Aguiar et al. 2015, which from the same sample of the same pool of Aedes aegypti mosquitoes used both full RNA-seq and small RNA-seq to look for viruses, and they showed that for both these methods, they find the same two viruses as being present in the sample. What they also showed is that what you can imagine using only these small reeds, that it can be quite difficult to make large contigs, which they actually managed to do. They actually managed to make quite some large contigs here. You can also imagine that if you have viruses, unknown viruses or very divergent viruses, that building contigs can become quite more difficult using this approach. However, and this is the clear benefit, if you look at the size reads and mapping to your viruses, for actively replicating viruses, you would expect this strong 21 nucleotide peak, indicating that these viruses seem to really be actively replicating inside your mosquito, which is what you are interested in. So now I want to talk a little bit about virus discovery that I've been doing in Culex pipiens in the Netherlands. And why am I interested in the Netherlands? One of the reasons is that I'm part of the One Health Pact, which is a large Dutch national consortium, which aims to prepare the Netherlands for future outbreaks of arbovirus disease. So I'm looking at insect-specific viruses in this specific context. And in the Netherlands, the most important mosquito species to look at would be Culex pipiens, which is the most abundant mosquito inside the Netherlands, and also the principal vector for arboviruses such as West Nile virus and pseudovirus. Pseudovirus has been circulating in the Netherlands for quite some years now. It causes outbreaks in blackbirds specifically and really strongly affects their population size. But Culex pipiens is also a main vector for West Nile virus, which causes outbreaks in southern and southeastern Europe, but also recently the United States. And in 2020, for the first time, a West Nile virus outbreak was detected in the Netherlands as well. So what I'm looking at is I'm trying to understand what the impact is of insect-specific viruses on arbovirus transmission in the Netherlands. And for that, we first need to know which insect-specific viruses are, of course, there. And actually, for this project, we started from scratch, as no one had really been looking before. For this, we started from 9 pools of Culex pipiens females. 3 of those pools came from Utrecht, which are actually the pools that turned out to be West Nile virus positive in the summer of 2020. And in that same summer, we also 6 pools were collected in Wageningen, a bit more eastern of the country. And to do virus discovery, we actually decided to use a culturing method. And the reason for this is that we not only wanted to find out which viruses are present, but we also wanted to obtain isolates for these viruses and really study their effect on arbovirus transmission. So what we did is we took mosquito homogenate, we added on Aedes albopictus C636 cells, which are immunocompetent cells and are widely used to culture viruses from mosquito tissues. And for 5 out of the 9 pools, we saw a cytopathic effect, which was a visual effect on those cells. They were either dying or their growth was slowing, indicating that there was something happening here. And for those pools, we took the medium, we extracted RNA, and we used nanopore sequencing to determine the causative agent of this cytopathic effect. What we found is that actually at both locations, we found a set of 3 viruses, which were alpha-mesonivirus 1 and negavirus, which from literature were 2 insect-specific viruses, but also umatillivirus, which is a virus that not only has been found in mosquitoes, but also in dead birds in the United States and also in Germany. So this seems to be an arbovirus, for which we actually know very little, and it would be very interesting to study this arbovirus more in the future. However, I continued working on these insect-specific viruses first. And one key question was, well, so I find these 2 viruses, but how important are they for the virome of the Netherlands? So from the same summer of 2020, I actually also had mosquito pools from 14 other locations. And initially, I looked at 3 different pools of mosquitoes from these locations. And these pie graphs indicate for each of those pools whether alpha mesonivirus 1 was present, nega virus was present, or both of these viruses were present. And as you can see that both these viruses are present either in 8 or 10 of the different locations. And based on the pool size, it seems to be about 1% of mosquitoes seem to be until now infected with these viruses. There's still much more. I would still want to look a lot deeper and also in other species, but at least it seems to be viruses that are quite prevalent. Alpha mesonivirus 1, it's a mesonivirus, which are actually very interesting. They are very large RNA viruses, 20 kb, positive stranded, enveloped. And one of the things, if I looked at my sequences, they were very similar to each other, more than 99.5% sequence identity between them. But then if I compare them to all known isolates of alpha mesonivirus 1, and really taking this whole cluster along, the identity between all of these was more than 97.5%. It was really only these two alpha mesonivirus 1 sequences here, which used to be actually called a different virus, where the sequence similarity goes down to 92%, which is still quite high. But it's really striking to see how all of these isolates across all of these countries have a genome that is so similar to each other. The story for NAGAF viruses was a bit different. NAGAF virus is very confusingly part of the genus NAGA virus. The genome is a lot smaller, but it's still a positive stranded, single-stranded RNA virus. And here, these isolates here were quite closer related than the isolates that I found. So the sequence identity between all of these was about 92% on the nucleotide level, whereas mine were clustering outside of there with about 85% nucleotide identity, with very, very good coverage. I do not think that this is enough to consider this a novel species. The amino acid identity is a bit higher, of course, even. So I'm not sure yet, but I don't, initially I don't think so. So I want to wrap it up here about this part. Like, I find these viruses in Culex mosquitoes inside the Netherlands. They're quite prevalent. There's a lot of different ways in which I can go. I specifically want to look at the effect on arbovirus transmission. I have isolates of these viruses, but there's many more things I could do, and I'd love to discuss that a bit more maybe after the presentation. Because now I want to zoom out. And of course, I'm not the only one doing research on insect-specific viruses inside mosquitoes. That would be a very lonely research field to work in. But while going through all these papers and reading all this research, a saying came to mind to me, which would be like, I can't see the forest for the trees. We have all these individual researchers looking at their individual species at different locations, and they're planting all these trees. And now I'm wondering, well, so how does the forest look like? How does it look like if we put all of this information together? What do we now learn about the mosquito virome? And this is really what we try to do with this work. And this is work that I've done together with two other PhD students from my lab, which are Jurgen Mone and Michelle Schinkel. And we co-published this together. If you look at the publications over time, you can see that the interest in the insect-specific viruses in mosquitoes is increasing. We basically we did quite an extensive PubMed strategy to find all possible papers that we could find that find a insect-specific virus in a mosquito. And you can see that this is increasing over time, which is specifically due to the increasing use of sequencing, which can either be RNA sequencing, small RNA sequencing, DNA sequencing, on mosquito samples. You can see that these mosquitoes have been caught all across the world, although there are some countries that are overrepresented, and that therefore a lot more studies have been done. So for these 175 studies, we wanted to know a lot of different things. It, however, boils down to 4 categories. We wanted to know which virus was found in which mosquito, at which location, using which technique. And ideally, and we tried to do this as much as possible, we wanted to know this for each mosquito sample that these studies had tested. And this is because we later wanted to do quantifications on the prevalence of these viruses, and therefore we wanted to retain this information if that was possible. One other decision that we made is we wanted to look at what these studies reported, and we specifically decided not to reanalyze metagenomic sequencing data for this purpose. We put all this information inside a database, inside an Excel database, which became 11,262 rows long, which had information about 1,289 unique viruses across 129 mosquito species in 57 countries. It looked approximately like this, where you have on the left side, you have the study. This guy is Botson Life 2021. The virus they reported, associated taxonomy at family level, and actually a separate tab on virus. And this is, we added this for the purpose of our review, because we wanted to ensure that whatever virus was reported could be linked to that same virus being reported in other studies. And because different namings, different spellings were used, we wanted to clean up this list to ensure that we could compare as much as possible. The second thing that we did, sometimes people refer to viruses like this. This is 3,110 Pertiti-like, which did not really unambiguously note one virus, and therefore we turned these into unknown Pertiti viruses because they didn't really associate with one unique virus. We also have information, of course, about the mosquito species. The location we tried to keep as specific as possible. So if the study said it was a different location, we also accepted this as a different location, which becomes important for a reason I'll tell you a little bit later. And all this other information about the mosquito sampling stage, blood feeding status, whether they were looking at RNA or DNA or something else. Now, for a lot of the questions we wanted to ask, now we had this database, we wanted to make quantifications on how often a virus or a virus family was in our database. How prevalent was it? How often have people reported it? And one problem we ran into, or we realized, was that because we kept this information per pool, if a PCR study from the exact same location would start doing PCRs on individual mosquitoes, such as is the case in here, you would get a lot of entries in our database, which don't really add a lot more information. And if we counted these as individual instances of this virus being detected, the amount of entries in our database would be quite inflated due to these studies, whereas they wouldn't really add a lot more information. To solve this, we started working with unique virus entries, where we filtered our database to ensure that each entry would at least be different in the study, the virus, of course, the mosquito species, and the specific location. So here it's quite important. This is not a country. This is if the same study finds it in 10 kilometers apart, that would be 2 different locations. This is how it approximately would look like if you indeed have this is a made-up publication where you have the same virus and the same mosquito species in the exact same location. It would turn into only 1 entry. Whereas if you have the same virus in different mosquito species or different viruses in the same mosquito species, you would retain this information. So now we could look at basically what viruses have been found. And this is compiling all of the information in our database and looking at it from a family level. And what you can see here on the right is that the largest group of viruses are actually unclassified. And this is not surprising because a lot of the effort, a lot of sequencing studies have been done in the last few years, and the taxonomy for these viruses just has not had enough time to catch up with this yet. And it also indicates that there's a lot that we do not know yet, and there's a lot of diversity that we do not quite understand yet that can be found in mosquito data. The second thing you can realize is you can see that some viruses are far more prevalent than others, and that you're also able to detect viruses from the microbiome, such as these phage families. Now, looking at this information, grouping all of the mosquito species together is perhaps not the most relevant. Therefore, we wanted to look at this information at a different level. The species level would, of course, be ideal, but for each individual mosquito species, there was little information. So we decided to look at the level of a mosquito genera. And this made quite some sense because 3/4 of all the publications reported viruses in either Culex, Anopheles, or Aedes mosquitoes, which were the 3 representatives I showed pictures of in the beginning. The other quarter were subdivided across 11 different genera, and each had much less information on them. So then basically what we looked at, we used a database to look at the virome of each of these mosquito genera, which are also mosquito genera that are very important for the transmission of pathogens, which is of course the reason why people have been sequencing them a lot more. So here I'm showing you basically how abundant, how prevalent each of the virus families, or the most prevalent virus families, in this case, in Culex mosquitoes. So we have a lot of information there, and which is to indicate some important variables to take into account. So first we have the number of unique virus entries, which is our measure for prevalence. But we also show the number of publications, which would say something about how often what it's found in one paper would also be found in others. We also show the amount of countries by the size of these dots in which these families have been reported in these mosquitoes, and an indication of how many viruses we're talking about, whether it is a family with representatives from a lot of different viruses, or perhaps there's only a few viruses that represent this family. And the key thing, of course, if you look at this graph, is you look at the flaviviruses, as they seem to be the most prevalent inside Culex mosquitoes. And this was not only the case for Culex mosquitoes, as the same thing was found for Aedes mosquitoes and Anopheles mosquitoes. So if you look at it like this, it really does seem to be the case that flaviviruses are the most prevalent viruses in these mosquito genera. However, if you look at the methods used to obtain these virus entries, you can see that a large reason of why these viruses are so prevalent in our database, because people have been looking for these viruses a lot more using biased strategies using PCR. But if you would not take these into consideration, you would still have a very prevalent virus family, but it's not clearly more prevalent than all the other than virus families such as Rhabdoviridae or Parvoviridae, for example. And this bias for PCR studies in flaviviruses makes a lot of sense. People do screenings for arboviruses, which also have some very important representatives in the Flaviviridae family, and then report also insect-specific flaviviruses. But also people, it is thought that flaviviruses, within the same family, the interaction between, for example, an insect-specific flavivirus or an arbovirus from a Flaviviridae family would be greater, and therefore there's a lot of interest in these insect-specific flaviviruses. But a point here also to make is that it's very important with such a database to keep into consideration which methods were used to obtain this, so you can keep these sort of biases into consideration. Now, if you look at the virome composition across these three genera, there is something you can say though. I already talked a little bit about flaviviruses, but the same, you can see actually that the same families are prevalent in all three mosquito genera. So we have flaviviruses, but also rhabdoviruses, which are negative-stranded RNA viruses. Parvoviruses are single-stranded DNA viruses, and also mesoniviruses. And the key thing to note is that these are all virus families where a lot of publications across many different countries find that these viruses are present in these mosquito genera. So there's quite some evidence that these play an important role in the virome. However, I do want to point out one thing, and that is the coloring for virus counts. You can clearly see that that mesonivirus is a lot lighter, meaning that there's less viruses that are representing this family. And that is unsurprising, because alpha mesonivirus 1 is by far the most abundant virus inside our database. Here I'm showing you a table with the most widespread individual viruses inside Culex. You have the virus, the family, the amount of countries that across the different continents that these viruses have been reported, amount of studies in which they were found, across how many mosquito species within the genera they were found, and the amount of unique entries. And for alpha mesonivirus 1, you really see that it has been found in a lot of across the world, and also in a lot of different mosquito species. But this is only the mosquito species inside Culex. If you take all of the database into account, it's been found in 34 different mosquito species across 5 genera, including Aedes and Anopheles as well. I do actually want to make a broader point though. So it's not only alpha mesonivirus 1, but if you take a look at all of these prevalent viruses in our database, you can see that they've all been found in multiple different mosquito species. And if you recall my introduction earlier, this is actually a little bit surprising. Because if you have the dogma that most of these viruses would be transmitted vertically, or that's what you would assume, there's actually no clear transmission modes from where these viruses can be transmitted between one mosquito species into the other. So seeing that a lot of different viruses have multiple different hosts indicates that other transmission methods perhaps are playing a more important role than we usually think of. So of course, this is not necessarily very new that these modes of transmission exist. For example, viruses have been postulated to be transmitted via contaminated water bodies or via nectar sources, for example, as well. But before compiling all this data, it wasn't so clear how prevalent it was that insect-specific viruses are present across multiple mosquito hosts. Now, I want to make one final point at the end. And that is something we realized while compiling all of this information for our database. We noticed that trying to extract all this information was actually very difficult and very time-consuming. We basically we're trying to extract from these studies, what did you actually find? And this information was usually scattered across the paper, hidden sometimes in the methods, and finding all this information was therefore not easy. And I want to give a small illustration of this as well, just a small example. For example, we have studies here that really give you all the information sometimes that you're interested in. These are studies that tell you, well, these are all the viruses we found across the different pools using these identities, so you can really say something about how closely is it matching to these viruses. You can know something about how many reeds, in which mosquito species, in which habitat. And this is very insightful, and for publication, it's amazing, but for researchers trying to reuse such data, it's quite difficult. And that is really the point I'm trying to make, is wouldn't it be great if there would be a standard inclusion of a table, for example, in the supplementary methods, where researchers can help you by saying exactly, well, per pool, we found these viruses in these samples, in these locations using these methods, and also giving you at the same time some idea of what this evidence is that they find these viruses. So also researchers can easily both understand and reuse the data because it's also in a format that people can use. And I'm really curious to hear from you guys and have a discussion about whether you find this, you'd also think that this is useful or not. And I hope to hear your opinion about this. Now I've reached the end and I have several takeaways for you. I've managed to isolate for the first time insect-specific viruses from Dutch mosquitoes, getting an initial idea of what is actually there, and being able to study these viruses, which is very exciting. We compiled a lot of information, and by doing so, we were able to say something about the family composition across different mosquito genera, finding these families that seem to be reported much more often than others, and that they are the same virus families across mosquito genera. And of course, this is a very broad view. If you really want to say something about the virus composition, you really have to sequence individual mosquitoes, which is something that has not been done before. But finding these broad patterns, I think, does help us to understand what we want to know and what we want to study in the future. The second thing is we find that insect-specific viruses seem to have a very broad host range, or at least a lot of them seem to have a broad host range, which says something about the way these viruses are likely to be transmitted. Finally, I want to propose a standardized reporting format, helping researchers to easily interpret and reuse data from metagenomic sequencing information without having to reanalyze the data themselves. That is what I wanted to share with you today. I have a lot of people to thank here. Specifically, I want to thank again Jurgen and Michelle, but also Pascal and Ronald, who have been supervising me, and also collaborators from the Wageningen, UNR, Leiden, and Erasmus, which were involved in sampling all of these mosquitoes that I could detect viruses in. Help from Bas Oudeminnik with sequencing, and also advice from Sander and Corbin about the projects in general. Finally, I want to thank my whole lab, which is depicted here, for being, I think, the best lab I could imagine working in. Thanks, everyone.
Thank you very much, Tom. That was a very clear and interesting talk. And while we're waiting for questions to come in, people feel free to raise your hand to ask a question or drop your question in the meeting chat. Yeah, so I'm actually part of the NFTI for microbiota, and we're building a virus database. And so maybe we should talk as well, because that's a lot of overlap. We're also interested in making standards for virus metadata and other microbial related data. So yeah, expect an email from me at some point.
Yes, please.
Or feel free to contact me as well. Okay, so maybe I'll start with the questions. There was one in the chat already by Kurt. He said, is there much risk of mosquito viruses becoming human pathogens once in a million years, or plausible?
Oh, yeah.
Yeah. So what is your perspective on that?
I mean, so basically the idea would be that somehow from human to mosquito to human transmission, you would change the transmission from human to human. I'm not quite sure. To be honest, I haven't given it much thought. I think it's relatively unlikely. I think the more interesting question would be, I always wonder, you know, if insect-specific viruses can ever add an extra host. And I was wondering if somehow insect-specific viruses could become arboviruses. Because if I go back to the introduction, right? You have a transmission cycle, but you kind of turn into a chicken egg problem of where it came from. So of course, the transmission of these viruses can change, but it doesn't seem very intuitive for me to go from a human to human transmission. But I don't have any specific arguments to back that up with. Shall I go to the next question?
Maybe. Okay, the next question I see is Stephanie Marot. She says, thank you for your very interesting presentation. Do you use a specific procedure to prepare cell culture supernatant before nanopore preparation and sequencing?
Not specifically no, actually. I mean, we use we do use Trisol specifically to extract RNA from liquid samples. But apart from that, we don't have a very specific protocol. That was enough for us to pick up these viruses, and that was our input for nanopore sequencing. So we don't really do anything specific there.
Okay. If anyone has a follow-up, also please feel free to turn on your microphones or put it in the chat. If not, I will go on to the next one. Okay, Marcus is saying, okay, how to define a new viral species? If 85% similarity might not be low enough, how low should it be? Are other tests, example, wet lab, required to define a new species?
I think this is actually a really good, I actually had a slide on this at some point. Wait, maybe I can find it. Because this is also a problem we specifically ran into. Oh, let me show it like here. So here, for example, I have a table here that we had part of the presentation where, because people can, if you find a new virus, you can give it a new name. If you find a sequence, you can give it a name. And this paper, for example, gave these names. But then if you look at the identity, it's vastly different. And you're wondering at some point, well, when is it this virus that seems to be with 96% amino acid identity seems to be that virus, and when is it not? And I think for now, there is no clear-cut cutoff that is defined for viruses to say, well, this is what is new. And for each individual's, for each different virus family, there can be different criteria. And at some point, these criteria will be discussed, and then people will say, well, this is the amount of amino acid identity that is needed. And then you go from there. But for a lot of different viruses, I don't think this is there's a, what do you call it? An accepted rule for this.
Yeah, I think this is a big issue. And of course, there's the International Committee for the Taxonomy of Viruses, and there's a whole, 200 people who are working on this problem, apart from all the virus researchers. But yeah, definitely to be debatable. Yeah.
And they cannot keep up with all this sequencing that we are doing. A lot of these viruses that we find, even viruses that have been found quite a lot, are still not taxonomy. We don't have a clear taxonomy for it or clear species definition. So yeah, it's difficult.
Yes, but in light of time, maybe we can go through some other questions.
Yes.
So the next one from Jorge. This ISV of flavivirus family, how much like dengue or other flavivirus pathogens? How much is it like dengue or other flavivirus pathogens?
As in gene, I actually don't know too much. I think the genome organization is quite similar, if I remember correctly. So I mean, I don't know percentages, amino acid percentages, or that by heart, but I think the genome, the composition is quite similar. But I could be mistaken here. I'm not. I don't know this very well. I'm not sure if there's someone here in the audience here who's working on this as well, who maybe has a more precise answer for this. But I wouldn't know.
Feel free to chime in.
Yes, feel free to chime in. Exactly. I see a question from Sebastian. Thanks for your presentation. I was curious about the methods you used to screen for the 3 viruses in Netherlands, not the ones you sequenced, but the others. I may have missed it. Was it PCR? Yeah. So we set up, I set up PCRs for these viruses And basically, I compiled all the current isolates for these viruses, and I looked for PCR primers that would amplify all of those, but none of the closely related, for example, Mesoni viruses, that would quite clearly not be amplified with this, to ensure that I really should be only amplifying alpha Mesoni virus 1 or NAGAT virus in that sense. And it's just endpoint PCR, by the way. Yeah. Next question. Do you know if there are mechanisms among ISVs to actively resist to the host RNA interference cleavage? Yeah, for sure. There are several ISVs that express proteins that interfere with the functionality of dicer, for example. And so these are things that people are looking into, and this is definitely something that has been that is known. Okay. That is more what I was thinking. Mozzie only thinks adding an extra host. Yeah, exactly. That's, I mean, it's again, I don't have any, I mean, I think for Bunya viruses, it's really the idea that they think that the ancestral Bunya virus would be an insect-specific virus, and then somehow it added this extra host. But for a lot of them, I don't think we know. And also for Bunya virus, we don't really know, of course. It's still an idea. Did you see any competition between viruses during the in vitro isolation as you used pools that can contain numerous viruses which can infect a specific cell line? Yeah, so we definitely had pools with different viruses in there. And I don't think I saw any strong reason for competition. The one thing I can perhaps note is, for example, some virus, like for example, NAGAT virus was a lot harder to detect on its own than the other two, which could be a problem with titers, although the titers seem to be very similar between the viruses also in those mixed pools. So somehow, I'm not quite, I'm purely speculating here, but it could be that there's some sort of interaction with NAGAT virus with some of the others, because this has also been reported a little bit in literature where they found that NAGAT virus seems to be present with, for example, a Mesoni virus. But this is a little bit more speculative in this sense.
So the next question is from Patricia. Yes. Patricia, can I invite you to turn on your microphone and ask your question?
Hi.
Thank you.
Hi, Tom.
Hi.
Thank you for your talk. Sorry. Hi, Tom. Thank you for your very clear talk. Very interesting talk. And I'm working at CIRAD in France on arboviruses with Culex pipiens. And I'm a little bit surprised that you didn't isolate more than 3 insect-specific viruses, because I did the same things you did on culture cell from pool, from Culex pipiens. And I have exactly the same result than you. Means alpha mesonevirus 1 and negavirus and Umatilla virus, but I have also more insect-specific viruses.
Oh, that's actually very cool. So we only find 2 insect-specific viruses, of course. I mean, it could have to do with the sensitivity of the sequencing, because I have to say, like, we had some problems with nanopore sequencing, where part of our library seemed to be formed by the adapters adapting on itself, which I think lost us a little bit of sensitivity. However, at the same time, if there's a virus really clearly replicating in my cells, I still think I should have been able to pick it up. But yeah, I'm not sure. Like, it could, of course, I mean, it's a different pool of mosquitoes in a different country, perhaps at a different site. We're not quite sure how variable the virome is across different habitats. It might depend where you find these mosquitoes. So there's, of course, a lot of different variables still.
We work in different habitats in rural area, urban area, and we find exactly the same. But the problem is to isolate more specifically one than the other one. And I have another question. Do you have an idea of the prevalence of the alpha mesonivirus 1 in Culex pipiens in Netherlands?
Yeah, so I think, I mean, I have to, because I have pools, right? So I have to do some smart calculations here that I have not done yet because I only recently did these. But I think based on, because the pool size is mainly 10 mosquitoes, I think if I do the math in my head, it's you know, like half to 1%. Okay. I mean, it's not super high, but still, I'd say it's quite prevalent. But I mean, I have so many more pools I could test, so I could probably tell you much more specifically in some time.
Okay. Thank you very much. It's very interesting, and that I think we have all people who are working on arboviruses. We have the same question, and what is the role, or what are the roles of insect-specific virus against arboviruses, and with the competence of.
Can I ask you one thing though? Like, how many viruses did you find? How much would you expect?
I work on a pool of 30 mosquitoes individual, and I found six different species of insect viruses. Okay. Yes,
that's very cool.
But first we did the NGS analysis from the pool, and then I did a PCR detection. And the problem is after it's to isolate, to perform competence, to identify the role of the insect-specific viruses. It's a little bit interesting because through different places of the world, in Europe, we detect the same virome.
Yeah, very interesting.
Yes. It's nice. Thank you for your talk.
Very welcome.
Thank you, Patricia. I think we have time for one more question to squeeze in. Jelle, would you like to ask it?
Sure. Can you hear me?
Yes.
Yes. Yeah. The question I had written down is, well, first, I think was a very nice presentation, very well, very clearly presented. So congratulations on that. And then the question I had was, you use C636 cell lines, which are Aedes derived, as you know, to identify Culex viruses. Did you consider using Culex cell lines to do the same experiment?
Yes. This is a good question. So we have two Culex cell lines in our lab, and also we have in Wageningen, there's another. So we have CT cells, we have HSU cells, and there's child ball cells that we have around. The downside is the HSU cells seem to be very resistant to viruses, and also already have an insect-specific virus present, Rida virus. And the CT cells seem to be a bit more easy to replicate viruses on, but they were infected with 3 other insect-specific viruses. So we chose this because we had a cell line that was very easy to work with because there was no insect-specific viruses present. But at the same time, we are working and have been able to get rid of some of these viruses from these cell lines. So I think in the future, we'll be doing this with Culex cell lines instead.
All right, great. And maybe quick follow-up questions. These viruses you are able now to isolate in the C636 cell lines, you're just able to propagate them multiple passages in the same cell line that works nicely?
Easily, yeah.
All right, great. And maybe a final quick comment. Well, two kind of comments. First one, I think if I understood correct in your one of your conclusion slides, you mentioned that sequencing of individual mosquitoes is not yet done. If that is what you said, then I think we have several studies where we looked at the viromes of individual mosquitoes. I would be happy to talk about those at one point if you're interested. And also the 2 last figures you showed, which you said is informative data, but not very useful if you want to do the analysis you want to do. They're also both from our group. And we will also, if there's other ways we can present the data in the future, we'd be very happy to do that in ways that might make your life easier. So very happy to work on that to solve that issue.
Thank you so much for this question. I mean, indeed, what I try to say about using, I'm very aware that people have been doing sequencing in single mosquitoes. However, the point I was trying to make that if you want to say something about compare the virome composition of mosquitoes across genera, you would have to do a lot of this sequencing to really say, well, this seems to be a virome composition for Culex pipiens mosquitoes, or for even Culex mosquitoes as a whole. So I meant to say that you have, there's a whole lot more we need to do before we can talk about virome compositions in such a broad way. And of course, look, I'm not necessarily saying that the way you represented it, I think it was a really good way to represent the data. For sure, was not my intention. I meant for the specific purpose of quickly finding out, hey, so what is the percentage identity for this virus? It was, of course, very difficult, and I don't think that's necessarily a critique on the paper. I just mean it as an illustration for if we want to do this more often, if we want to look at this virus data in this way that we did, it would be great if we also have an extra table where all this information is compiled together. So definitely don't see that as a critique. I think it was
no. I didn't take it as a critique, but I think you make a valid point that it's not always that easy to get all the exact data you want. But usually all the raw data are present in SRA, so there are ways to get back to it, but it's quite computationally intensive, of course.
Thank you so much.
Thank you very much, Jelle. I think I'll leave the call open for like 7 more minutes or so because we have still some questions. And why not ask them now? So Maxim, could you please ask your question? If you would like. No response. Okay, maybe I will ask his question. Why did you choose to use nanopore sequencing?
Yeah, it's a good question. We mainly chose to do nanopore sequencing because we figured it would be much quicker to get the answer we needed. We didn't necessarily expect a very complicated virome in these cells growing out, especially after already 2 or 3 passages. And nanopore sequencing is just a lot quicker and cheaper to do. So that's why we wanted to do it with nanopore sequencing, because this would be an easier and quicker way to get the answer that you need. So that's why we decided to use nanopore sequencing.
Okay, thank you. Nicola has a question about the databases. Nicola, would you like to ask your question?
Yeah, you're talking about the database and how useful it could be. I think that's very, very useful. Do you plan on actually making something where someone can go in and find like an interactive version of lists and papers, or is this just an idea you think people should work towards?
Yeah, this is a question I get quite often, actually. It's, I mean, currently, indeed, the database is just an Excel file, and it's, to be honest, it's very, if you have some basic Excel skills, it's very easy to do. You can just filter through things and get some information quite quickly. But I mean, having a user interface would, of course, be much nicer and quicker and better. But this is not something, I mean, this is something that I personally don't have the time and resources for to keep that, and I'll keep updating this as well. I will say, though, I think maybe in the future, I mean, it would be great if these type of data could be somehow embedded in something like together with the NCBI resources, because I can quickly show this. I was very happy, for example, if you look at NCBI virus, right? You can quickly just put in a virus and see where it has been found. These type of things, of course, we could also do with our database. And it would be extremely nice to do, but somehow combining all that information inside of a resource center that has the money and time to keep curating and keep these things up to date would be ideal. But I think we are not going to do it as of now, at least.
Thank you.
Thank you, Nicola. I also put something in the chat. If you are interested in learning more about virus databases, we're actually having a workshop about virus databases at the ICTV meeting here in Jena, which is also hybrid, so you can join online. And I put the link in the chat for you to register. It's free to join. So yeah, everybody who's interested in databases and automating virus taxonomy, those are all topics that are going to be covered there. So definitely check that out. And thank you, Tom. Okay, I think I'm going to wrap it up because I don't see any more questions in the chat. Thank you very much. And thank you, everybody, for joining today and for your nice questions. So with that, I think I will close the session. Thank you.
Thanks, everyone.
Bye-bye. See you next
Bye.
time.